The next speaker is, the first speaker is Gerlinde Metz, who can come here. He's coming from Canadian Center for Behavioral Neuroscience in the University of Lethbridge. Thank you very much. And you will propose a talk on ancestral exposure to stress epigenetically programs, preterm birth risk, and adverse maternal and newborn outcomes. So you have the floor for 55 minutes. Gerlinde?
Thank you very much, Mr. President. Dear ladies and gentlemen, first of all, I would like to thank the organizers for inviting me here. I'm truly pleased to be here, and it's a great honor to have the opportunity to present some of our recent data and discuss further about transgenerational inheritance. I'm really glad that I'm following these excellent speakers that we have listened to before, so that I can dive right into the topic of stress. And I would like to start off with a question to you. Who of you would know your great-great-grandmother? I don't see many hands. And who of you would think that your great-great-grandmother, so 4 generations removed from us today, would still have an impact on your personality and your well-being and lifetime health today? So this is a topic that I would like to discuss with you. I will talk about an animal model using rodents, mainly rats. And probably when you even listen to any news today, open the newspaper this morning, you probably encounter some effects of stress that are highly populating the media. So you read headlines such as "Mother stress depression affects unborn child, severe distress accelerates aging," and so on and so on. And so this indicates that stress is indeed a very, very strong, very salient influence on our health from early development to old age. So we're looking at the lifespan health profile. We're looking a lot at phenotypes that we were discussing before. And we're also combining this with genomics, epigenomics, and metabolomics. So it's actually not very unlikely that a mother, and I'll be focusing on mothers, but you can account for similar mechanisms playing a role in the health of fathers too. Actually, it's not unlikely that mothers during pregnancy would experience stress. Stress is unavoidable pretty much these days. And actually 80% of women report that they experience some mild to moderate or even severe distress during their pregnancy. So we would like to understand what are the implications for the baby and even potentially subsequent generations based on this maternal stress experience. So stress, as we've heard before, generates a footprint that travels from one generation to the next. It's being passed on through the generations. And it will influence adaptation of the offspring to a certain environment. It will influence the degree of stress resilience, how the offspring, how the baby, the child later on, and the adult offspring will deal with the stress, will deal with food scarcity, food abundance, and so on. It's a very important programming that takes place that enhances survival later on in life. But also there are some detrimental consequences to the experience of especially adverse programming by stress. And we'll talk a little bit about this and the benefits of it today as well. So we hypothesized when we set off with our project that stress in ancestors would program maternal and offspring health trajectories. We were very interested in the risk of preterm birth based on a team project that got funded. We were also interested in newborn health outcomes, and then we let these animals age. So we ended up having 5 generations of aging rats that we paid $1 a day per animal throughout many years. So in our hypothetical framework, we're assuming that the prenatal environment is very important, experience in the womb that interacts with postnatal environment and that also interacts with heritable genetic and epigenetic factors to all together affect the maternal well-being and well-being of the offspring. In our session today, we've heard a lot about heritable genetic and epigenetic factors, and these indeed play a very important role in livestock production and breeding. So I think the mechanisms that we'll be discussing and that we've heard about today apply to probably a lot of vertebrates that matter to our research. So in my work, we're focusing on rats. And rats show a great deal of maternal care. You might be familiar with the work of Michael Meany and Moshe Ziv about maternal care. And first of all, we were interested to see if prenatal stress would affect the offspring's health and the risk of preterm birth. So this is a behavior that we identified just for the purpose of these studies. This rat is highly pregnant and is expecting her litter to come on board very soon. So what she's showing here is a normal behavior, which we call tail chasing or rotational behavior in her home cage. This behavior was recorded at night with infrared cameras. And this tail chasing behavior, we assume, is a practice for later on when the pups are being born to retrieve the pups to the nest. So this is an indicator of postnatal, postpartum parental care, maternal care shown by the mother. So we were very interested in this behavior by a prepartum animal, the dam, and you see that tail chasing was mainly observed in normal, healthy, not experimentally stressed animals. As soon as the mothers were stressed, they did not show much of this behavior anymore. So obviously, our dams were indeed stressed by the mainly psychological stressors we're using during the last week of gestation. But what we did not find was our expected change in gestational length. We did not find any linkage to potential risk of preterm birth in stressed mothers, which had been reported in the human population. So we started also to look at the offspring. We looked at prefrontal cortex, which is highly involved in stress response regulation and mental health. And we found that in the offspring, stress did have an effect, for example, in behavior, but also in gene expression patterns in the prefrontal cortex. We found transcriptomic changes relating to cell signaling pathways, structural brain structure, oxidative stress, immune response, and so on. And about 80 of these genes were involved, documented to be involved in neurological and mental health issues in the human population. So let's take a step back to the mothers. We are very interested in epigenetic changes. We did DNA methylation profiling, but we also profiled the microRNAs. And we're very interested in microRNAs because they make very good biomarkers. There are a lot of these microRNAs that have been demonstrated to be linked either causally or as a symptom to human diseases. And so microRNAs are short, non-coding RNA molecules that actually interfere with the gene expression at the post-transcriptional level. So they interact with the mRNA molecule and block the protein assembly. So microRNAs can target many different gene products actually, but what we were interested in is microRNAs related to later health trajectories. So for example, in our stressed mothers, we found in her brain, her prefrontal cortex, microRNAs being altered that are linked to brain plasticity, apoptosis. And these were downregulated. That means that was a release on the conventional understanding of microRNAs, that they serve as suppressors of gene expression that would release gene expression for their target genes if microRNAs are downregulated, which we saw induced by gestational stress in our dams. So potentially some of these microRNAs are heritable to subsequent generations. So what we did then, to see maybe if we find other patterns in maternal health, we bred more generations of our animals. We bred 4 more generations up to the F4 generation. So this is the model that I'll be talking about in my next slides. We had 3 main lineages. We had an unstressed lineage with no experimental stress in each of the generations. Then we had a transgenerational stressed lineage, in which only the F0 parental generation was stressed, and then their daughters, great-granddaughters and great-granddaughters were not stressed during pregnancy anymore. Then we had a multi-generationally stressed lineage in which each of the dams was stressed again. This simulates a population living in a constantly stressful environment, such as for humans we could envision poverty, food scarcity as being a very long-term stressor. So we were comparing these different lineages. And how about preterm birth now in our several generations? Well, let's take a look at the F1 and F2 generations. The daughters that were born to stressed mothers actually did show a variation in gestational length. You see the blue bars on the left that indicate non-stressed parental animals. The parental generation that was stressed also did not show, as expected, did not show higher risk of shortened gestation, but their daughters in the F1 generation and even their granddaughters in the F2 generation, no matter what stress lineage they were derived from, they showed significant shortening of their gestational length, down to about 13 hours. If you shorten gestation for much longer than this, actually you yield non-viable offspring in rats. So there was a significant risk of preterm birth. We also found that ancestral stress led to elevated levels of blood glucose in the F2 generation. You see here on gestational day 18, and then after parturition on lactational day 1, there were elevated levels of basal glucose in the F2 generation, especially in the multi-generationally stressed lineage. We've looked at many other parameters, but also we've looked at microRNA expression patterns again. So Carol Menderson's lab in the US has actually established a very discrete pathway of regulating preterm birth risk. And a central parameter, a central pathway here is the microRNA 200 family. So the MIR200 family in our profiles from all the microRNAs that we looked at was the one that was most significantly changed in the uterus. So here you see that in the F1 and F2 generations, there was an upregulation of microRNA expression. Potentially, assuming these are repressing gene expression, they would downregulate their gene target expression, which is exactly what they did, what you see here. The MIR200B family regulates ZEP2 and STAT5B in the uterus. Both of these genes are very important for pregnancy maintenance. If you reduce their levels, there's a higher risk of going into labor. And this is what we saw in the F1 and F2 multi-generationally stressed population. There was a downregulation in those two genes and a number of others that are involved in preterm birth. This pathway was derived from humans. Obviously, we could assume that there's a similar mechanism also working in rats for parturition. So what is interesting is that if we look at epigenetic markers, we would like to have a proxy. We'll talk a bit more about health trajectories. We would like to know how we can predict later health risks. And, for example, readily accessible at birth is the placenta. So we were comparing placenta with uterine gene expression and microRNA expression patterns, and also brain expression patterns. And what you see here is an example of our findings showing that in the F2 and F3 generations, the gray ones indicating cortical expression of a microRNA involved in brain development, and the placenta in black of the same microRNA, that their correlation is actually closely connected, that a high level induced by ancestral stress in the cortex is mimicked or preceded by a high level of microRNA expression in the placenta. So obviously, this gives hope that the placenta could be a way to potentially predict some of the health risks that might come on board later on in life. And this is something that we would like to follow up further. So how about the offspring? if we know a bit more about the microRNA changes that obviously took place in the offspring and in the brain and in the placenta, we also saw changes in their behavior. For example, one of the tests we did as early as postnatal day 7, when the pups still have their eyes closed, we placed them on an inclined plane, head facing down. And the pups would like to be on top of things, so spontaneously they start turning around and walk up the inclined plane. This is what you see on this little movie clip. The pup was placed head facing down on the plane. It starts turning around, and we measure the response, how long it takes the pup to realize they're being placed on an inclined plane. This is a sensory motor response, and it's a very sensitive developmental milestone for pup development. What you see here is on the y-axis the time that animals face down, and you see it broken up for each of the generations. You see that the prenatally stressed offspring, the yellow F1S bars, actually show needed twice as long to realize they're being placed on the inclined plane to turn around. Then with each generation, F2, F3 generation, in the offspring, we found that there was an increase, a gradual increase in their time to respond to the stimulus. So there was a delay in their development, in their brain development. Let's take a further look at the F3 generation. An interesting test that we did for lifespan health, we did, for example, on the multigenerationally stressed cohort, where we monitored motor activity as the mean distance traveled that you see here, from early adolescence to old age. 1.5 months is an adolescent rat. 3 months, 7 months is the height of reproduction for a rat. So at 3 months, the boys that we looked at would be going out looking for a girlfriend. Then at 13 months of age, this is an older rat. So what was interesting is that we're interested in motor activity, for example, which is highly referenced in the human population. Maybe there's an indicator that ancestral stress could play a role. At least in our adolescent rat pups, we saw elevated motor activity levels when they were adolescents. And then again, when they were older, at the height of reproductive age, there was no difference between normal non-stressed and stressed animals. So there are obviously very vulnerable periods in life when we can see the effect of ancestral stress being displayed, which is early development and old age. We let these animals age, and I'll show you an example of a disease pattern here. We looked at the onset of diseases in our animals. When they were about going close to 2 years of age, we found a higher incidence of certain diseases in our F4 generation. You have to imagine this is the transgenerationally stressed cohort where only the great-great-grandmother was experimentally stressed, and then there was stress no more. So in this population, we found that there's a higher rate of renal failure, a higher risk of tumor cancer formation, and a higher risk of lung disease based on the experience of ancestral stress at the age of 2 years. Renal failure and lung disease, in this case pulmonary edema, both indicate high blood pressure, which was obviously programmed by the experience of stress in the ancestors. So we're following up more with biomarkers that could predict these diseases earlier on in life as we have tissues collected from this population. So this all sounds like pretty gloomy news, and we've talked about positive experiences. And here I would like to give you an example, giving hope that beneficial experience at any time in life can actually overcome some of this ancestral programming. And we used enriched environment in adolescent rats and housed them in this environment where they have many friends to play with. They get different types of foods on a daily basis. They have many different toys that are provided to them on a regular basis. And so they're very healthy, happy rats. And we housed the ancestrally stressed cohorts in this enriched environment. This is what we found. You see an example on the left-hand side of animals housed in the enriched environment, and below you see the standard housing that is usually used in laboratory environments. So we measured the stress response in our animals. We also measured a whole lot of other responses. Here you see the basal plasma corticosterone, the main stress hormone in rats, at basal levels. And you see that across the board, the enriched animals in the red bars showed lower levels of the stress hormone corticosterone in basal conditions. Also, their stress response was much healthier when they actually were encountering an acute or a chronic stressor. So overall, these animals were happy, healthy, laid-back animals. And we indeed found that some of the microRNA profiles and DNA methylation profiles that we found to be linked to ancestral stress were actually partially reversed by this very positive experience. So I would like to summarize that ancestral stress is very important to prepare future generations. Just think of the motor activity. I think being very active in a stressful environment enhances the survival in this environment, enhances the chance to escape from a predator or from a potentially other stressful situation. It's a very positive effect, and it leads to ancestral preparation by stress could enhance resilience to these situations or metabolic preparation. But these changes come at an expense. There's a price to pretty much everything. And here we have the price tag attached to this adaptation, in that there's a higher risk later on in life to cardiovascular diseases, metabolic changes, and accelerated aging, as well as also a greater risk of preterm birth for the young mothers that we looked at. So ultimately, we would like to turn this around and learn about transgenerational programming of health trajectories by looking at biomarkers, genomics, epigenomics. We look at urine metabolomes, we look at blood metabolomes and solid tissues in various different ways in order to find predictors early on in life that could tell us who's at a greater risk of a disease, maybe 10, 20, 30 years down the road. So ultimately, our research is dedicated to being able to predict and hopefully one day help to prevent some of these conditions. I would like to be optimistic. So for building the foundations for healthy futures, I think it is important to recognize the importance of transgenerational programming and their impact, its impact on lifetime health trajectories. And epigenetic mechanisms do play a large role in some of this preparation and adaptation and some of the adverse health effects that we've been seeing. And also I'd like to point out that this is a very important model to learn more about disease prediction and prevention. So I would like to very much thank my hardworking lab and my wonderful collaborators at the University of Lethbridge, but also at other universities, University of Alberta and McGill. And I would like to thank you for your attention and for listening and attending this session. And I will be happy to take any questions.
Thank you, Gerlinde, for this fascinating talk and very optimistic also conclusions. I like very much that. So do you have any question for Gerlinde? Yes, please.
Hi, I'm DJ de Koning, Swedish University of Agricultural Sciences. Thank you very much for a very inspiring talk. Some recent research in humans and mice from Jonathan Flint's group showed that prolonged stress has a very strong effect on mitochondrial reproduction. There's a lot of mitochondria being produced in result to stress, and telomere length. Now, is that another type of effect you could try and factor into your predictions?
Thank you. I think you're raising some very important issues. We actually plan to look at telomere length, which is associated with accelerated aging in human populations. I think this is a clinically very valid marker. And the mitochondria dealing helping to deal with prolonged stress in terms of producing more energy for cell survival, I think is a very important factor. We haven't looked at it in our experiments yet, but these are certainly on the radar screen. And thank you.
Because in that, sorry, very quick comment is that in his work they showed very easy ways to look at mitochondrial yield using very low coverage DNA sequencing.
Yes, these are really excellent comments. I think it's a very complex phenomenon we're looking at here. So we have a very complex pathway, a cascade, leading down to cellular functions. And what we've seen so far in our metabolic output from cells, actually that significantly altered, which might be linked to mitochondria function. Some of the markers we found are indirectly linked to mitochondria function. So this is a really important future aspect of this research. Thank you.
So next question, please. Yes.
Thank you for the fascinating presentation. You talked briefly about...
Could you give your name and origin?
Oh, I'm sorry. My name is Sophie Igelen. I work for CRV, which is a cattle breeding company. You talked briefly about epigenetic effects resulting from an interaction between the prenatal environment, postnatal and heritable effects. Now, in dairy cattle, we use embryo transfer quite a lot. So you have a different heritable ancestry, or you have a different ancestry than the prenatal environment. Is there anything known, or are you aware of any studies that have looked at which factor plays a larger role, or if there's anything known about the effects coming from embryo transfer recipient?
This is a really great point. Thank you for raising it, because indeed early life is very important. There are studies on DNA methylation patterns based on preparations of embryo or in vitro fertilization and so on. So there were actually gene imprinting pathways linked to later higher risk of certain diseases. I think based on the enrichment studies, though, that there's a potential that the postnatal environment could somehow favor some more beneficial developmental trajectories. Maybe not make it up for all the previous experiences, but I think there is a very high chance that some of the adverse consequences could be at least mitigated to a certain degree.
Thank you.
Thank you very much. This will be the last question for this talk. Thank you very much, Gerlinde.